Welcome, everybody, to topic 5 in marine ecology, and we're talking now about adaptation in the broad sense to the marine and aquatic environment. We're still at the level of the organism, asking how the interaction between organism and environment has guided the evolution of traits of the organism. And we've been talking about social traits, mating and social behavior, then about life history traits last time, and how life history traits are integrated to maximize fitness. And so now we focus on conditions and resources within the marine environment and how those conditions and resources may have guided the evolution of populations in general. And this is going to be a bit of a review for some of you who have taken the marine biology course in Zadar, but we'll add a few details here and there. So most of this is going to be familiar to you already. All right, so this is a huge topic, an endless topic, and one of the reasons that people get interested in marine biology to begin with. And so I posted some textbooks, some great textbooks on the Merlin page on marine biology and marine ecology that you can check out for more details. But we're still at the level of the organism, and now we look at the maintenance or survival portion of the life cycle, just the day-to-day activities that an organism needs to carry out to be functional and happy, mainly focused on moving around, finding food. And as I said, this topic is basically infinite. And so, in one lecture, one weekly topic, we can only look at some broad categories, basically tip of the iceberg. So we're focusing now on the maintenance portion and all the wild and weird and wonderful adaptations in this portion of the life cycle. So we're looking at Darwinian selection and Darwinian fitness. And so we're right straight here in the middle of Darwin's struggle for existence now. And we're focusing on conditions and resources and how they may have affected the Darwinian fitness. So remember, conditions are properties of the environment that influence fitness of organisms that cannot be consumed. So conditions include factors like temperature, salinity, light intensity of different wavelengths, pressure, currents, tides. Resources are quantities in the environment that can be consumed to increase fitness and that you can compete with other organisms for. So these include food, including other organisms living or dead, nutrients for plants, and space for plants and animals. It can be used to escape predators, or it can be used by predators to search for and ambush prey. So the kinds of conditions and resources in the environment can shape the evolution of any species or population. Often you can see predictable relationships if you know what the conditions and resources are. And at the center of these topics is the idea of the ecological niche. And there have been several definitions of the term, but the one I will highlight right now originated from Hutchinson. So the basic idea is that for a population to maintain itself, the birth rates must exceed the death rates for any time interval, at least equal the death rates, equal or exceed. So we can define a niche as the area in the environment in which conditions and resources allow the population to exist and maintain itself. So as a niche is not a habitat, so we need to distinguish it from a habitat. Habitat is just a specific kind of location in space and time. A niche, rather, by the Hutchinson definition, is a multi-dimensional hypervolume within which the population can maintain itself. We're considering conditions, the range of conditions of all types within which birth rates are at least equal or greater than death rates during a time interval. So, for example, the niche of this shrimp, this bay shrimp, maybe it can maintain its population within this range of temperatures on one axis. and within this range of salinities on another axis. So outside those pink bars, the population decreases. At the pink bar, the population growth rate is 0. Within the pink bars, the population growth rate is positive. So the idea that is that the pink bars here represents the limits of the condition within which the population can maintain itself. So outside, the population growth rate is negative, so the population decreases. So you end up with this volume within a condition space that represents the niche of the species. And we can add any number of additional conditions on new dimensions. So we don't have to just have 3 dimensions, in which case we get, this nice volume here, representing the ranges of salinity, temperature, and pH within which the shrimp can maintain its population. pH is important, remember, from climate change and the ocean acidification, as is temperature, of course. Okay, so this is the idea of the niche, n-dimensional hypervolume. Originally proposed by G. Evelyn Hutchinson as his concluding remarks in a symposium in the US. And there's no limit to the number of axes you can add to this plot, so we don't necessarily need to stop at 3. We can add axes and have these hypervolumes that we cannot visualize because we can only see in 3 dimensions. So the concept of niche is related to the ideas of function and structure in biology. You need to have a particular function and a structure to be able to exist within this niche, and different niches necessarily are going to be requiring different types of functions and structures. So the conditions and resources within the species niche are those that guide its evolution over time and are the conditions and resources that the species has become adapted to. So relating to the ideas previously, all species are absent from almost everywhere on Earth because each species is functionally and structurally adapted to a particular niche, and trade-offs have reduced the adaptedness of each species to other conditions and resources. So each species occupies this n-dimensional hypervolume within condition resource space. And that volume is a set of environmental factors to which the species has become adapted. So all these concepts come together and interact in this concept of the niche. So we can look at the function and structure of each species and relate those to the niche it occupies, understand that function and structure as possible adaptation to those conditions and resources. So there are 2 types of niche defined. These are called the fundamental and the realized. Just a conceptual distinction. So the fundamental niche is the volume of conditions and resources ranges within which it can maintain a population in the absence of enemies. So for example, competitors, predators, parasites. That's the fundamental niche. If there are no enemies, then the species can expand throughout its fundamental niche. The realized niche, which is smaller in volume, is the volume of conditions and resources within which a species can maintain its population in the presence of enemies who might prevent it from maintaining a population in some areas of its fundamental niche because they are adapted to those areas and lower the fitness of the species enough to prevent it from maintaining a population. Okay, so we've got a predator that occupies this range of the conditions. It might prevent the species from existing and maintaining a population in that region of its fundamental niche. In the absence of the predator, then the species can expand into that region. The presence of predator, the species is restricted down to this realized niche. So the realized niche might be quite small compared to the fundamental niche in the presence of the full array of all the competitors and predators that a species needs to figure out how to make a living with and around in the community that it interacts in. So today we'll look at a sample of the full range of adaptations that marine species have shown to the major conditions found in the marine environment. So the major gradients are in light, salinity, temperature, oxygen, density of the water, pressure as a result of that density at depth, the substrate, the ground underneath the water column, and the nutrients and food that are available as resources in any particular location. So first, let's look at the major conditions and how they vary in the marine environment. So marine environments are divided most coarsely according to depth and proximity to land. So depth is correlated negatively with light penetration and positively with pressure, both of which have resulted in adaptations of marine organisms. So the pelagic environment in this graph is the open ocean water. So the pelagic is divided into the neritic and oceanic regions based on distance from the continental landmass. Neritic is open ocean above the continental shelf. Oceanic is the open ocean beyond the continental shelf. The pelagic zone is also divided according to depth. The euphotic zone is the surface zone, which is defined by light penetration high enough to permit net positive photosynthesis or net positive carbon fixation in plants. The dysphotic is the area just below the euphotic, light penetration enough for some gross photosynthesis, but not enough for net photosynthesis. The respiration rate here is higher than the photosynthesis rate. So the net photosynthesis is gross photosynthesis minus the respiration rate. The depth limits of euphotic and dysphotic are variable, and they're based on light penetration, which depends on water clarity, which can be really highly variable. Below the dysphotic is the aphotic, which extends down to the sea bottom. So the aphotic is defined as a region receiving less than about 1% of the incident light. And then the aphotic is divided into 3 zones. That's the bathypelagic, which extends from 1,000 to 4,000 meters in depth. Then the abyssopelagic goes from 4,000 to 6,000 meters. And then the hadal zone, which is below 6,000 meters, 6,000 meters of the sea floor. This is the deepest region of the ocean, where the Greek myth of Hades resides. These are the deepest trenches, no light, very high pressures. The benthic environment is the sea floor, which consists of sediment and rock of various composition and origin. The continental shelf is the extension of the continental landmass, and the seaward end of the continent is the end of the continental shelf. It usually is a gradually sloping surface that is less than about 200 meters depth in most places. So the continental slope is a slope beyond the continental shelf. It's a steep landmass beyond the shelf, and the continental shelf is considered a trench when it reaches depths below about 4,000 meters, especially down below 6,000. It is a trench. The continent below the neritic zone, right here, this is called subneritic. And the continent below the oceanic zone is called suboceanic. That's this portion. So suboceanic here, subneritic here. And this suboceanic zone is divided into the bathyal, which corresponds to the bathypelagic region of the open ocean. So underneath the bathypelagic region, the ground there underneath that part of the ocean is called the bathyal zone. And then we have the abyssal zone here below the abyssal water. And then the hadal zone here below the below 6,000 meters depth. And then the area of the benthic region subject to tidal action between the high and the low tides has its own terminology that is different in English and Croatian, and possibly other languages, I don't know. So you need to understand these terminological differences. So in English, the region between high and low tides is called the intertidal region, or less often called the littoral zone. Littoral. Intertidal and littoral mean the same thing in English, the region between high and low tides, which can be narrow or wide depending on the resonance properties of the ocean basin and the distance from the amphidromic points, as we saw in the oceanography course. The region above the high tide that's nevertheless influenced by seawater, usually spray from the wind, is called the supralittoral zone. So this is above the high tide, but it's still different from a terrestrial habitat because it gets salt spray, especially during storms. In Croatia, during a bura or a jugo, salt water will blow up onto the land and create a unique habitat up there, a unique niche also. And the region below the low tide is called the subtidal in English, or the sublittoral zone. Subtidal or sublittoral zone, and that's divided into the inner and the outer continental shelf. Just divide it in half. So all of this is subtidal. It never is exposed to the air. So it's called subtidal or sublittoral, and inner versus outer continental shelf in English. And here's some more diagrams of the English terms. And I want to make a distinction between the Croatian terms, which are different from the English terms, even though they're similar words. They mean different things. Okay, so you'll need to be familiar with this difference in Croatian versus English. Very confusing. So the intertidal zone in Croatian is called the mediolittoral. That is the intertidal zone. In English, this would be called the intertidal zone or the littoral zone. The littoral zone. In Croatian, it's the mediolittoral. The supralittoral zone in Croatian is the same in Croatian and English. Only in English we put two T's here. So supralittoral is the same in Croatian and English. The sublittoral zone in English is the infralittoral zone in Croatian. So in Croatian, they say infralittoral. In English, we say sublittoral. And the continental shelf below the sublittoral zone in Croatian is called the circa-littoral. Continental shelf below the sublittoral zone. And there's no English term corresponding to this except maybe subtidal, but it's usually not used for areas far from the intertidal zone. So in Croatian, the entire continental shelf is included with the littoral term or zone and consists of supra, medio, infra, and circa-littoral zones. So in English, the supralittoral here would be supralittoral. The mediolittoral would be the littoral or the intertidal zone. The infralittoral would be the subtidal intercontinental shelf. And the circalittoral would be the subtidal outer continental shelf in English. So the littoral term in English only refers to the area between the high and the low tides. Here we have another diagram from a local book written in Zagreb in marine biology. The Croatian terms. So the basic differences here in Croatian described. So here's a nice map of the continental shelves of the world. Most of the continental shelves are here in the far north. And the Adriatic Sea has nothing but continental shelf. There is no slope. You have to get into the Mediterranean and into the Atlantic to see the deeper water. And in English, to further the confusion here a little bit, we also have the term fringe. So you've got here the intertidal zone right through here. So we have the upper intertidal and the lower intertidal. And above the intertidal, you can divide this zone into two separate areas. One is called the superlittoral fringe, and then the actual zone a bit above that. That's called the spray zone, where you got salt spray up there that creates a different niche from a terrestrial or marine environment. And then we also have the fringe applied below the intertidal. So this would be the infralittoral fringe. And then below that, we've got the subtidal or infralittoral zone in English. So fringe is just an extra habitat area that's sort of a transitional from the intertidal upwards or the intertidal downwards. And there's also a term here called eulittoral, which means the middle part of the littoral zone. All right, let's first look at adaptations within the pelagic zone. Pelagic, remember, is the open ocean zone, which is divided up into different subzones based on depth. So we've got the euphotic, dysphotic, mesopelagic, bathypelagic, abyssopelagic, and the hadopelagic zones that we need to consider to get a brief sampling of the adaptations in the pelagic zone. So pelagic species consist of passive drifting plankton and active swimming nekton. And plankton can move upward or downward in the water depending on density of the water, density of the organism, and resistance of the organisms to the water. Okay, so those relationships are described here in this equation. So S here is the speed of movement, the speed of vertical movement in this equation. dW is density of the water, d is density of the organism, r is resistance of the organism to the water, so that would be the surface area and shape of the organism, primarily contributing to the r here. And V is the viscosity of the water, which depends on the temperature and salinity. Higher temperature or lower salinity means lower viscosity. So the relative density of organisms in water determines whether the organism moves up or down, while the product of resistance and viscosity determines how fast it moves up or down. Resistance is also called drag in English. The more resistant you are, the more you feel the water is dragging against you as you move through it. So this graph of salinity against temperature can be used to find the density of water. For example, a salinity of 33.5 right here and a temperature of 9 degrees right here results in a water density of, on this graph it says 26.0. What that means is the density is 1,026 grams per liter. So as temperature increases or salinity decreases, the water density decreases. And these lines are lines of constant density. So if you know the salinity and temperature, then you know the density of the water, and you can compare that to the density of the organism to know whether the organism, the plankton, is going to sink, is going to move downwards in the water column, or upwards. Pelagic organisms increase drag or resistance with their body shape or structure. For example, parachute structures increase drag as high surface area, also flat or long projections, and joining with others to form a chain formation. These all reduce the speed of movement through the water. Many of these structures, these feathery structures that increase drag, these flat or long projections in the plankton, are very beautiful, wonderful um, morphology, and they were the subject of paintings by 19th century artists, such as our friend Ernst Haeckel, who coined the term ecology, started this field. And there are many others who followed suit with these beautiful paintings and drawings, such as this German artist who painted these specimens from the Gulf of Naples. Some plankton are active and exert energy to stay in the same place or move up or down. Krill, for example, which are shrimp-like zooplankton, a major food item for baleen whales. These, for example, beat their pleopods to maintain their position in the water. So they don't move much horizontally, but they do maintain their vertical position and are able to do that with their legs. So these beautiful high surface area structures are interpreted to solve some problem relating to a drag or resistance and to assist with maintaining position in the water column. So look at these two examples. Do you predict plankton with higher or lower surface area in warm water? Which is the warm water species and which is the cold water species here? So medusas, or we call them jellyfish in English, or jellies for short, have a parachute-like body adaptation form that can contract and relax to move up or down. There are many examples of chain-forming species in the plankton, where several individuals will join together to form a chain, such as colonial tunicates or salps, salp like this one here, which form flat, long ribbon-like bodies consisting of several individuals fused together, sort of teamwork. Chain formation is common among diatoms. Right here, the previous is another chain forming tunicate. This is a diatom, and these have bristle-like extensions that increase resistance and help to maintain position within the water column. And this is another chain former, several cells joined together in a chain of different individuals. Okay, buoyancy is determined by the relative density of water and the organism, which is the numerator of the equation for S here, which is the speed of vertical movement of a planktonic organism. The denominator is the product of resistance and viscosity of the water. So this buoyancy can be modulated by changing the density of the organism in several ways: by fats and oil production, by gas production, and by bodily fluids, the chemistry of body fluids, heavy versus light ions. So fats have a lower density than water, and they also provide good insulation, so they have a dual purpose. Gas can be added inside cells or inside organs, like, for example, fish have swim bladders, or trapped in external attachments on seaweeds, like floats. Also, several species control the chemistry of their body fluids. Sulfate and magnesium are heavier ions than ammonium or chloride ion. So you can change the chemistry to alter your buoyancy and position in the water column. In diatoms, we have siliceous spines made out of silica and a large surface to volume ratio. They form chains. They can also accumulate fats, and they control their ions. So they use many methods to control their density, and they move up and down in the water column in the day versus night. During the day, they're up in the water column to take advantage of the light, and they photosynthesize. And then at night, they move down lower in the column. Bacteria in aquatic habitats have gas vesicles or bladders or vacuoles inside their cells. These are organelles inside single cells. And their function seems to be to modulate buoyancy. So you can move towards the light at times when light is available and move out of it when it's not available. So cyanobacteria have vacuoles that they can change the volume of by turning on and off genes. And they can destroy the gas vesicles if they want to increase their density by physical methods such as pressure, turgor pressure against them. In addition to vacuoles, they can also change relative amount of the dense and less dense ions to further regulate. And all of this can be, you can use light as a stimulus in various ways. So seaweeds such as sargassum, which is a macroalga that drifts passively on the water surface, um, it's not attached to anything. It just is free on the water surface, and it forms what's called the Sargasso Sea in the Atlantic within the Gulf Stream current, and it has floats, these bladders filled with carbon monoxide gas, and these bladders help keep it at the surface of the water and exposed to maximum illumination by the sunlight. And why would they be filled with carbon monoxide instead of carbon dioxide? Well, maybe to deter grazers on the flesh of the seaweed because the carbon monoxide is toxic. So we use the term neuston and plouston to describe plankton that exist right at the surface, right at the interface between air and water. For example, there are species such as the Velella velella. It's a famous cnidarian, also called by a common name by the wind sailor. It has a sail at the surface, which causes the animal to move along the surface at a constant angle to the wind. So if the wind blows in a certain direction that causes these organisms to move towards the shore, you'll get this huge stranding of thousands of these Velella velellas up on the beach. You'll see one day the beach will be clean, and the next day, after a storm, maybe you'll see thousands of these washed up on the beach. So the Neuston is a very specialized habitat that appears to be useful because it takes advantage of wind currents to move the species where it needs to be. At least that's the hypothesis. It might be also a useful place to be because you're relatively separated from predators that are deeper in the water column. So you're up near the surface where you might be more safe from predation. We have the Portuguese man of war. This is also a gas-filled float that maintains the body of this animal at the water surface. It's never in the water column, it's right at the surface. And it has these long tentacles that you've probably heard of that are very dangerous to come in contact with, long stinging tentacles. And the Portuguese man o' war is another example of a colonial animal, an example of amazing, incredible teamwork. It consists of many different organisms. Each one is called a polyp, and they're working together. They've divided the labor. There's the floater, also called a nomatophore. That's a single organism. Then there's the predatory organisms. Those are the long tentacles, stinging tentacles. Each one of those is a different, separate organism. Then there's digestive organisms that break down food, and then reproductive organisms that make the gametes, the sperm and eggs, and they all combine together to form one functioning animal. Amazing example of teamwork here. Or we could call it cooperation. And all of these organisms that make up a single Portuguese man of war animal, they're all genetically identical. They're all clones of each other. So their relatedness is 100%, all helping each other out, altruistically helping each other out. So that was a quick introduction to some of the adaptations of the plankton, the neuston. Now we move to the nekton, which are the active swimming members of the pelagic zone. And there are many kinds: fish, marine vertebrates, cephalopods, which are squid and octopus, marine birds, turtles, sea snakes. These all have adaptations as well for regulating buoyancy. So in cephalopods, nautilus, sepia, and spirula, they all have chambered shells filled with gas, and these provide buoyancy. Squid does not have such chambered cells. Rather, their tissues have a high content of ammonia, which reduces buoyancy. They do have an internal shell that you obviously will find when you clean these. And you also find their internal shells washed up on the beaches often. In fish, there's a swim bladder in some bony fish. The swim bladder contains gas, which increases buoyancy. And some species can add or remove gas from the esophagus quickly, and other species can add or remove gas only from the bloodstream. Sharks have high fat content in the liver, which increases buoyancy. You can buy shark liver oil as a dietary supplement. So no swim bladder, but high oil content. In the sperm whale, the spermaceti is the pearly white wax-like material in the head cavity of the sperm whale produced by the spermaceti organ at the top of the whale's skull. So the whale can regulate buoyancy by heating or cooling the spermaceti. Heating it increases buoyancy. So this oil was primarily the reason the sperm whales were hunted, basically for that oil, but now it can be obviously substituted by various different types of oils, plant oils like the jojoba oil, which is taken from the seed of the jojoba plant, which is about 50% oil. Okay, so the necton has many different ways that they move from place to place underwater. It's just an amazing diversity of methods for locomotion, that many of which engineers have used. And there's a great video, and I will link to that to avoid any copyright problems with this one. So check out the video. In the video, we see a medusa using a pulsing bell. Comb jellies have beating cilia. Siphonophores, they have a pulsing nectophore. Pteropod sea butterflies, they basically flap their wings, fly underwater. Squids, they flap their fins and they jet. Dumbo octopus has pulsing arms and fins. Deep sea octopus, pulsing arms and jetting. Hatchetfish has tail fin power. The gulper eel has a sort of a whip-like tail motion. The rat tail has an undulating tail, and the white skate and other skates, they hover using their fins. So we have sea cucumbers use body contractions. Sea spiders use striding legs. They sort of walk through the water. It's amazing. Deep sea spiders, backward leg propulsion, and the feather star has alternating arm strokes. And you'll need to take a look at the video. When you see the video, then you'll have to believe what I'm saying. It's amazing to see all these. So the squid can move around by flapping their fins, and also they trap water inside the mantle cavity here, and then contract muscles to jet it out through their siphon, and that can give them very rapid, explosive movement, which in cephalopods is called jetting movement. They jettison the water, so in English we call it jetting locomotion. Fish generally move as a result of undulating movements in the body and tail from side to side, as opposed to up and down in the cetaceans. And this is achieved by alternate contraction and relaxation of the myomeres, which are the W or V-shaped muscle fibers in the body and tail. The myomeres are what you eat on your plate. And the shape of the fishtail is a compromise between maneuverability and speed. And the two extremes are slow moving but easily maneuverable. They can move around tight spaces easily. That's one extreme, but at the cost of having to be slow moving. And the other extreme is high moving, fast moving, but not easily maneuverable. So the progression is rounded tail to truncate to forked to lunate to heterocercal in the shark, and that's the progression of tail shape from high maneuverability and low speed to low maneuverability and high speed in the shark and swordfish. Schooling seems to be an adaptation both for feeding and for avoiding predators. So apparently swimming in a school can increase fitness in some species because it increases the probability that any individual finds food or avoids a predator, because you might not see food or predator, but individuals nearby might see it and alert you. So there's an advantage to everybody to hunt and move about inside a school. Nevertheless, many species hunt or avoid predators while solitary, and this is a question that's not really fully answered. It's not fully known why some species school and others do not. But it's easy to imagine what the advantage of schooling could be. And the pinnipeds, which are the seals and sea lions and walruses, the movement method depends on the group. So in seals, like this typical seal, genus Phoca, the hind legs are turned permanently back into a pseudo tail, and they cannot rotate these hind legs forward. It's not possible while on land. So on land, they move like an inchworm. In water, they propel themselves using their pseudotail, sometimes their forelimbs also, which can be used either for steering and maneuvering or for a little bit of propulsion in addition to the tail movement. In sea lions, hind limbs can rotate forward. It's a major difference. And they can support weight. So they can gallop on land like a horse. So they can move on land much, much more efficiently and faster than seals can. And in the water, they propel themselves primarily using their forelimbs, which have greater musculature and bigger bones on these forelimbs. Manatees and dugongs are slow-moving herbivores, and they propel themselves with up and down undulations of their tails, which are broad and paddle-like, similar to whales. And they use their forelimbs primarily for maneuvering. Cetacean propulsion in all species is via undulating up and down movement of the tail flukes. So in these guys, the flippers are used mainly for maneuvering. All right, let's take a look at adaptations relating to temperature. Temperature profile in the oceans generally, that profile is called a thermocline, and there's a permanent thermocline below about 200 meters in depth. So as you go deeper, the water curves along a predictable curve. So the thermocline is up here, permanent, and then you have seasonal thermoclines to shallower levels. So in the spring or summer, it gets warmer up near the surface. And so you get a stable surface layer of warm water. And just below that stable surface layer is a rapid shift from warm to cooler temperatures. And that shift is called the thermocline. And then during the winter, you've got a surface mixed layer, which is cooler. And then below that, you have the permanent thermocline that is present all year round. Notice that in deep waters, the lowest temperature is what? Is 4 degrees Celsius. That's the lowest temperature in the deep waters. That's because 4 degrees is the temperature of the maximum density of water. So the water at the bottom of any body of water can't be any colder than 4 degrees. If the surface temperature is 4 degrees or higher. The main adaptations to a changing temperature, a variable temperature in the water, are mechanisms to regulate body temperature. So mammals and birds maintain a constant body temperature of close to 37 degrees, no matter what the external temperature is. And this temperature is maintained by heat produced during energy metabolism. Large fish like mackerel and sharks and tuna can also maintain a body temperature above around 30 degrees, regardless of the external temperature. And most other vertebrates and invertebrates have the same body temperature as water temperature, and this includes most fish and reptiles. And 35, 37 degrees is really close to the maximum temperature that you're likely to see in surface ocean waters near the solar equator. The internal control of body temperature to 30 to 37 degrees in most species that are able to do this is called endothermy, and endothermy has a cost. The cost of burning sugars metabolically to produce heat. And this cost can be measured as the rate of oxygen consumption. When the temperature decreases below body temperature, respiration must increase to maintain the body temperatures. So that causes increased energy consumption and oxygen use as the external temperature goes below this zone right here. Increased oxygen use to maintain the body temperature up in this range. Species that don't maintain constant body temperature but rather have the same body temperature as the ambient surroundings are called ectotherms. So in ectothermy, there is no metabolic cost. The oxygen consumption is close to zero no matter what the external ambient temperature is. So endothermy results in high activity and fast movement regardless of the water temperature, but at a cost of energy, high energy oxygen consumption. Ectothermy, on the other hand, has the advantage of low energy consumption, but at a cost of slower movement in cold water. And that slower movement is a disadvantage if you're a prey trying to escape a predator in cold water and you're an ectotherm. So this slower movement by ectotherms in cold water has led to this hypothesis, this ecological hypothesis called temperature-dependent predation success. So seabirds who dive underwater to catch fish are called pursuit divers. They propel themselves with feet, for example, cormorants, or with their wings in auks and murres. And there's some nice videos. Will be linked at the bottom. So you can check this out. Pursuit divers occur only in colder water, average temperatures below about 15 degrees, and at latitudes higher than 15 degrees, so closer to the poles than 15 degrees. And this is apparently because their fish prey move more slowly in colder waters. So you don't see this pursuit diving in seabirds close to the equator, but you see it abundantly close to the poles, and it's amazing to see seabirds basically moving at such high speeds underwater, catching the more slowly moving fish, ectothermic fish, in the cold waters of the far north or south polar regions. All right, we already talked about adaptations in whales and dolphins and porpoises for diving in lecture one, but I just wanted to show one aspect we didn't discuss much, namely the dense mat of capillaries that surround the lung alveolus. And that dense mat allows cetaceans to very efficiently extract oxygen from their lungs, which is then stored in the blood hemoglobin and muscle myoglobin. So at the surface, they will take several deep breaths, and in these capillaries will charge their blood with the oxygen from those several deep depths, and then they often will expel the air from their lungs when they dive, and then use the oxygen in their blood and muscles during that dive. Let's consider light now. Light penetration in water drops rapidly with depth. 45% remains at 1 meter, 16% at 10 meters, and only 1% at 100 meters. The wavelengths that penetrate the farthest are those in the blue-green bands. And by 100 meters depth, the only light wavelength is in the blue portion of the spectrum. Light penetration also follows an exponential decay curve according to this equation, which you will use in your lab exercise this week. And the critical parameter is the attenuation coefficient K, which determines the rate at which light intensity drops with depth. And because light does not penetrate deeply, all photosynthesis occurs near the surface of the ocean within the euphotic zone, which extends from the surface down to a depth of about 100 to 200 meters, depending on how clear the water is. So photosynthesis combines carbon dioxide and water to produce carbohydrate and oxygen, sugar and oxygen. Carbohydrates are broken down to produce usable energy during the process of respiration, which requires oxygen and produces the products carbon dioxide and water. So the carbon budget of any plant depends on the rate of both photosynthesis and respiration. Photosynthesis takes carbon out of the environment and fixes it in the plant tissues, cells, and respiration puts the carbon back into the environment. The net photosynthesis is the net carbon flow into the plant, which is positive if the photosynthesis rate is higher than the respiration rate, and it's negative if the respiration rate is higher than the photosynthesis rate. So the bottom depth of the euphotic zone is the depth at which there is no longer any net photosynthesis. In other words, the carbon taken up during photosynthesis is exactly balanced by the carbon used during respiration. So below this, in the dysphotic zone, the net photosynthesis is negative. And above it, in the euphotic zone, the net photosynthesis is positive. And we'll have a lab exercise this week that makes use of these concepts. Photosynthetic organisms can be conveniently categorized as attached or free drifting. So phytoplankton, by definition, is free drifting. Such as this compound microscope shot of various species of diatoms. But it's an enormously diverse group from over a dozen taxonomic classes. And phytoplankton exists at all latitudes and in ocean, brackish, and freshwater environments. This is just a listing of some of the major groups, taxonomic survey of the marine phytoplankton. So it's important to understand the basic mechanics of photosynthesis, which in all photosynthetic organisms follows a saturation or parabolic curve with light intensity or carbon dioxide concentration on the x-axis and rate of photosynthesis here on the y-axis. So it will always be a saturation curve or a parabolic curve depending on the effect of light at high light intensities. So we're looking at light intensity here on the x-axis, and net photosynthesis is 0, where the photosynthesis rate equals the respiration rate. And the light intensity at this point, right here, where the curve comes down and hits 0, this light intensity is called the light compensation point. There's just enough light to compensate for the amount of respiration so that the photosynthesis rate has a net of 0. So in many cases, a very high intensities of light actually damage the photosynthesis apparatus in the cell. Then the photosynthesis rate falls. And this is called photoinhibition of photosynthesis. So like all saturation curves, the maximum has a maximum point, and that is the maximum rate of photosynthesis possible. And then the half maximum point is achieved at a light intensity equal to K, which is the Michaelis constant for this process. And as we talked about before, light intensity penetrates the water following exponential decline, which is a straight line on the logarithm scale. So this is the logarithmic scale up here on the x-axis for light intensity, and this is depth. So this is a nice diagram showing how light intensity attenuates as the depth of the measurement point increases. So the intercept of the line where it crosses, where it crosses the x-axis here, which is the light intensity axis, or the log of light intensity axis, this intercept of the line is determined by the level of incident light. So if the level of incident light is high, then your intercept is up here at the high end. For example, if you've got a sunny sky, you start here and then you decline from there at some rate depending on how clear the water is, which is determined by the light attenuation coefficient. So the light attenuation coefficient is basically the slope of this line, or 1 over the slope, depending on what the axes are. So here we have two lines that have the same clarity of ocean water, the same clarity of the water in these two lines. And we know it's the same because their slopes are the same. So the attenuation coefficient is the same. But one line starts out with a much lighter conditions at the surface, and the other line starts out with much darker conditions at the surface. So by the time it reaches a very low light intensity, it's a much shallower depth than this other situation with under full sunlight. So the euphotic zone extends down to about 1% of the incident light intensity. So that's 1 in 100. This is 5 going from 3 is 2 orders of magnitude. That's 1 in 100. So phytoplankton can operate here with a net positive photosynthesis in this region right here of light. Okay, so how deep this zone is depends on the attenuation curve. So if you've got clear coastal water under full sunlight, then that zone extends down to this depth right here. If you got the very clearest ocean water, then that extends down to a deeper depth. In this case, around 200 meters depth. So effective color vision can occur down to a depth of 1 times 10 to the minus 8th of the incident light intensity within the dysphotic zone. And the aphotic zone starts at about 1 times 10 to the minus 15th of the incident light. And this is about the light detection limit for deep sea fish. This right here, around 10 to the minus 10 of intensity relative to bright sunlight. So these deep sea fish have a very bizarre appearance, and many of their structural and morphological characteristics are not well understood, but others can be hypothesized to have resulted from the lack of light for the most part, and also the high pressures at depth. So these fish often have a very large gape, which allows them to ingest other animals larger than themselves. And this makes sense because the productivity, the amount of biological material produced by growth and reproduction at depth is very low, which necessitates long periods of time during which animals have no food and have to fast. So they're adapted to ingest fish that are very large compared to their body size and can be even larger than their body size. These fish often also have bioluminescent lures which attached to their heads and lure prey close to their mouths. And in some cases, species have no functional eyes, which makes sense because eyes require energy to produce and maintain. And therefore, individuals not wasting that energy because eyes are useless if there's no light, and the energy must be wasted then. And animals not wasting that energy apparently have higher fitness because they could put that energy to other more effective uses. Odontocetes, which are the toothed whales and dolphins, have evolved the ability to echolocate to find their prey. Of course, echolocation is very useful if you're a predator of mobile, fast swimming fish, and especially in water with low light penetration. So, sound is generated in the blowhole of the dolphin, the frontal air sac of the sperm whale. And this lens tissue in the forehead focuses the sound, or the spermaceti in the sperm whale. And the sound reflects off the front surface of the skull. And then sperm whales dive deep, typically half to 1 kilometer deep, and feed on giant squids, which they catch in very low light using echolocation. And this is well into the dysphotic zone. Deepest dives have been over 1,200 meters into the aphotic zone. Sperm whale, frontal air sac squeezes the air through the trachea And the clicks are generated here at the monkey's muzzle, and they travel backwards, reflect off the skull, and then are focused through the spermaceti towards the object that the sperm whale is interested in. It might be that giant squid. Which will fight back. All right, let's look at the benthic environment, the sea bottom environment. Most benthic biomass, which is the mass of living material, is in the coastal areas on the continental shelves. And the major division of habitats in the benthic zone is hard versus soft substrate. So hard bottom, we're talking about rocks. Soft bottom, we're talking about small particle size of sediment, sand and gravel, mud. Rocky intertidal habitats are the most familiar hard substrate benthic habitats. They're what we see when we go down to the beach. Rocky intertidal shows very pronounced zonation in animal communities from low to high on the shore. And in general, the higher biomass and diversity to be found are at low levels. So at the lower levels of the intertidal and then also into the subtidal. Okay, so higher biomass and diversity down here, lower biomass and diversity up here. This is a typical rocky, looks like a basalt bedrock shore on the west coast of the US. So the conditions at the rocky intertidal are challenging. So you have drying out at low tide, and if organisms can move to shelter or withdraw into shells, that helps. Having thick skin to prevent water loss or covered with rocks or shell fragments. And some species can biochemically tolerate drying. Another condition is strong wave action. So flexible holdfast in algae can tolerate the high energy. The currents can be up to, say, 20 meters per second, which is pretty high, and you need some kind of structures to deal with that. So you have strong glue, like epoxy glue, to attach to rocks, hard outer structures, shells, behavior of clustering together. So barnacles often, juveniles often settle where there is already a colony of adults. So as the tides go in and out, there is extremely variable chemistry. So the salinity varies, the temperature varies, the pH, oxygen vary. So a lot of species behaviorally respond to that. They withdraw into shells when the tide goes out. They have biochemical adaptations, for example, heat shock proteins and natural sunscreens, which help deal with the situation when the tide goes out. Competition for space is a big factor in the rocky intertidal. There's often lack of any space, primary space on the rock for attachment. Larger species can overgrow and undercut and smother smaller species, and smaller can attach to the bodies of the larger. And we expect the higher an organism occurs on the seashore, then the more specialized they are to those conditions when the tide goes out. More adaptations they have to desiccation, lack of oxygen, higher temperature, lower salinity if the tide happens to be out when it rains. And those species that are adapted to the higher levels thereby can then use those levels as a refuge from predators in the lower intertidal that are incapable of surviving those conditions, that are active mainly in the water and not exposed during low tide. So rock is an important habitat for marine algae. They're attached to it. And by being attached to it, that allows them to have a stable surface to prevent mortality. And also the water flows against their bodies, which allows a nutrient exchange, which would not happen as efficiently if they're free floating. So being attached and stationary means that they have currents flowing across their bodies all the time, which allows them to photosynthesize more efficiently. However, in order to use that rock, they need to be able to grow up to the surface in the lighted waters to photosynthesize. If the rock is too deep, then they might not be able to do that. But in areas where the rock does extend down in shallow places and where the algae can grow long enough. However, in order to use that rock, they need to be able to grow up to the lighted surface waters in order to photosynthesize. So the deeper the rock is, the greater the distance, the vertical distance they need to grow. So kelp beds are areas where the rock is in the subtidal region, and the kelp has to grow to very large sizes to reach the surface towards the limit of their growth ability. And so kelp beds are in subtidal areas where large brown algae, capable of growing very rapidly, like several decimeters per day during the summer, to reach the lighted surface. These form dense populations and provide structure and food for a diverse animal community. So kelp beds occur in the temperate zone in both northern and southern hemispheres. And these are the major genera: Laminaria, Macrocystis, and Nereocystis. On the east coast, the west coast of the U.S. Also, Laminaria is here. So the main species are in these genera. So the canopy kelp generally have bladders and floats, we talked about already, filled with carbon monoxide that hold their photosynthetic tissue at the water surface where incident light intensity is the highest. So those are the canopy kelps. The so-called shrub kelps are understory species adapted to lower light, and both types occur in the temperate zone. So the shrub kelps are sort of like shrubs in a temperate forest. They're underneath the canopy trees, also adapted to lower light conditions. So these kelp beds are an important component of hard substrates in the benthic zone of the ocean, close to the shoreline. We've looked at the hard substrate. Now let's take a look at the soft bottom habitats within the benthic zone. And these are areas of sand, gravel, and mud, small particles that can be excavated, can be dug up, easily dug up, and burrows formed. So first, let's take a look at the myofauna. Myo means sand, and fauna means animals. And myofauna are animals living on the sandy bottoms whose bodies are roughly similar in size to grains of sand. So there are many phyla of animals, including cnidarians, crustaceans, flatworms, nematodes. Small size might evolve to protect from many larger predators, such as fish, but myofauna are nevertheless consumed by their own predators, and also predators, worms that ingest sand, just basically swallow sand and then digest off any organic material in the sand, while then passing the clean sand out of their gut. So if you pick up a handful of wet sand and look with a magnifying glass, you'll see these small animals the size of the sand grains. And then you can also filter them out of the water in the lab and concentrate them and put them under a scope and get a much better look at them. Nematodes, flatworms, many crustaceans, polychaete worms. Macrofauna, on the other hand, are species much larger than sand grains, and most macrofauna of the soft benthic habitats are burrowers and include clams, polychaete worms, shrimp, heart urchins, and sometimes snails that are capable of burrowing into the sand. So, burrowing is an adaptation that allows the animal to avoid variation in water conditions above the surface and also hide from predators in the water column. But there are also predators in the sand itself, often Nereid worms that can swim through the sand when it's wet enough. So they've got predators in the sand and predators in the water column above, mainly fish nipping at the siphons as they extend out of the sand, waiting at the siphons at the holes, and then nipping the siphons as they extend to feed. Burrowing can create problems in attaining oxygen, especially in the mud. The sediment particles are very fine, they can block the flow of oxygen. So these also have adaptations for low oxygen, sometimes special types of hemoglobin that enable them to be aerobic for longer periods of time um, when there's low oxygen in the water in their burrows. Macrofauna, burrowing macrofauna also showed zonation from low to high tide levels, reflecting their degree of adaptation to exposure at low tide, just like on the rocky intertidal. More marine species occur in the lower areas, generally have a higher biomass and diversity in the lower areas. Truly intertidal species occur in the intermediate areas, and mostly terrestrial air breathers with marine origins, though, are on the upper beach. So burrowing is accomplished similarly in most soft-bodied marine benthic species. There's a digging appendage, and that appendage has a wedge function that enables it to penetrate soft sediments, and an anchor function as well that holds the forward part of the digger in place while the rear part contracts muscles to pull the body forward. In this lugworm, Arenicola, the digging function is performed by the head end itself of the worm's body. The head is the anchor and wedge, with the body segments flaring to provide additional anchoring function. Clams have a specialized digger organ which performs both the wedge and anchor function. The shape changes to a sharp penetrating form, allowing it to extend downward into the sediment, then expands to create an anchoring function. Then the digger muscles contract, pulling the body of the clam downward as the end of the digger is anchored in the sediment, and then it repeats. In hard-bodied species, on the other hand, most species' legs are used for a shoveling function. For example, the mole crab on sandy beaches. And the mole crab is also sort of oval shape, which tends to equalize the forces on its body, making it harder for the wave forces to pull the body out of the sand and make it vulnerable to predators. All right, so oxygen is dissolved in the water. If an animal is burrowed in sediment, it can deplete oxygen in its immediate burrow surroundings. So the primary adaptation to low oxygen in sediments is the evolution of a snorkel-like structure that extends from the body of the animal to the surface of the sediment, where it sucks in well oxygenated water and brings it into the gills of the animal. For example, a hard urchin here has a respiratory funnel that extends to the surface. Clams have siphons that extend to the surface, which function both to bring in food items and bring in oxygenated water. Another adaptation to low oxygen in the sediments is the evolution of legs in crustaceans that function as paddles that can create a current of water through the burrow. And this is fairly common in burrowing shrimp. So these legs in females can be used to create a current of oxygenated water around their eggs. And they can also be used to create a current of water between two openings of their burrows, so that you've got well oxygenated water always circulating in the burrows. So no matter how deep they are, no how muddy they are, and stagnant they are, you've always got this current of oxygen coming through. So no siphon, but a current coming through. In some cases, for example, in lugworms, the burrow is actually very shallow, just a few centimeters below the surface, and the worm has a special type of hemoglobin that is capable of storing large quantities of oxygen. That's called oxyhemoglobin, for example. In flowering plants growing into muddy sediments where there's no oxygen in the sediments, they need oxygen at their roots in order to pump ions in or out of across the cell membranes, especially roots in highly saline water, because high salt concentrations can be toxic. So the plant needs to pump out sodium and chloride, and this requires energy, which requires respiration, which requires oxygen. And this is a problem in salt marshes worldwide. And they solve this problem of no oxygen in the muddy sediment with a special type of tissue called parenchyma. And these are dead cells that are rigid, but empty and filled with air. So these cells extend from the roots up to the shoots. So these parenchyma are similar to the snorkels of some animals. Oxygen can diffuse rapidly in the air within the parenchyma from the surface down to the roots. So that's, for example, salt marsh plants whose upper portions are actually in the air, and then their roots are down in the muddy sediments with low oxygen. These parenchyma are absolutely necessary for these plants to survive. Adaptations to variable salinity are also absolutely necessary in intertidal regions and in estuaries. The salinity can vary at any location from basically full seawater to zero if the tide's out and it's raining, or if you've got a stream, freshwater stream coming in when the tide is out. So it depends on river and tidal flows at the time. Species living in estuaries, so it can divide it into four groups, depending on what they're tolerant of in the estuary. And these are the stenohaline, euryhaline, brackish water species, and freshwater species. So stenohaline are those that can survive only in water that is nearly 100% marine. Euryhaline species are those that can survive in a wide range of salinities, but not in complete freshwater. Brackish water species are true estuarine species that cannot survive in either complete freshwater or complete marine water. And freshwater species can survive in freshwater and water with low salinity. So these are freshwater species that just manage to make it a little ways into the estuary where the salinity is still, quite low. And in most estuaries worldwide, the number of species in intermediate salinities is small. The vast majority of species are found in close to marine water or close to fresh water. The fewest species occur right at 5 to 10 parts um, per thousand salinity. So these are, for the most part, brackish water species that occur only in estuaries. Species deal with salts in the water through osmoregulation, which is a process of pumping dissolved ions across cell membranes to maintain internal fluids at optimal concentrations of essential ions. So there are different types of species that tolerate variable salinity. So osmoregulators maintain blood salinity at a constant value, regardless of the salinity of surrounding water. And these are analogous to endothermic species adapted to temperature. So these osmoregulators can function across a range of salinities as long as the external salinity doesn't change rapidly. So, example, freshwater eel or salmon, some crabs. Osmoregulation is often not complete. For example, crabs can maintain high blood salinity until the external salinity, external salinity becomes quite small, at which point they can no longer maintain their blood salinity. Osmoconformers have blood that is close to the salinity of the surrounding water throughout the salinity range, and they still survive and function within some range of salinities, not necessarily all salinities. For example, estuarine polychaete worms, shrimp, clams, and most brackish water species. And there are certainly trade offs involved here. Osmoregulators have the advantage of stable internal environment, which allows fast and efficient movement, but this comes at an energetic cost, the cost of maintaining ion concentrations within a stable range. So it's a similar cost to endothermy. Osmoconformers, on the other hand, have lower energy requirements, but at a cost of an unstable internal environment, which places limits on ease of fast and efficient movement. So many of these species are slow moving and sedentary, such as many burrowing polychaete worms. They're just in the sediment, they just don't move much. Calcium is an important ion in seawater. It's the fifth most common ion in seawater. Many groups of marine animals use calcium to build their shells and their exoskeleton. So, the abundance of calcium is a tremendous useful resource for marine organisms, and they've evolved to use it for this reason. For example, corals have a calcium carbonate skeleton. The theca produce new calcium carbonate at their base, and they add continuously to the basal plate. The result is a production of new limestone and coral reefs. The result is a production of new limestone and coral reefs, so around an oceanic island. Corals produce a limestone fringing reef that over millions of years becomes a barrier reef as the island landmass erodes into the ocean. And eventually the island erodes completely away, while the coral reef remains as a ring around the island. So this ring of living, growing reef is called an atoll. However, the increasing carbon dioxide in the atmosphere is reducing the concentration of carbonate ions in the ocean, making it more difficult for organisms to build calcium carbonate into their shells and skeletons. So this process is called ocean acidification. The ocean is not acidic. It has a pH of about 8.06. pH of 7 is neutral. Because the ocean's pH is higher than this, that means the ocean is alkaline. And ocean acidification is a process by which the ocean is becoming less alkaline as the ocean absorbs CO2 from the atmosphere. It's actually not becoming acidic, it's becoming less alkaline. So over the last 200 years, the oceans of the world have absorbed roughly half of all anthropogenic CO2. And so the CO2 is dissolved in the ocean water. And once it dissolved, it reacts with water to form carbonic acid, H2CO3. And then carbonic acid then releases hydrogen ions, which lowers the alkalinity of the water right here. So the hydrogen ion concentration has increased by about 30% over the last couple centuries and will push the pH down to about 7.8 units by 2100, according to the IPCC, the Intergovernmental Panel on Climate Change. So the problem is these hydrogen ions bind to the carbonate right here to form bicarbonate, which has reduced the concentration of carbonate near the surface of the ocean by about 10% over the last century. So the lowered carbonate makes it more difficult for animals to form calcium carbonate necessary to build shells and skeleton. And it also increases the rate of dissolution, dissolving of solid calcium carbonate. So if carbonate concentration falls too low, the shells become soft and they start to dissolve. So here's a CO2 increase in the atmosphere and a dissolved CO2 in the water. And as the CO2 dissolves, the pH has been going down, becoming less alkaline. So the question now that is currently unanswered, do animals in general have enough heritable genetic variability in their ability to produce calcium carbonate to evolve adaptations to the falling pH of the oceans and maintain their ability to synthesize it and incorporate it into their shells and skeletons? Or will we see a widespread loss of these species over the next century? Currently unanswered, and it's a huge topic of research right now, looking for heritable variation in calcium carbonate biochemistry in these species to find out if they might have that variation necessary to adapt to the ocean acidification, which is happening very rapidly. All right, food capture. Probably the most common means of food capture for marine organisms involves simply filtering out plankton from the water column. So this method is called suspension feeding, just feeding on things that are suspended in the water column, particles that are suspended in the water column, this dilute soup of ocean water. And this method is used in some species in all phyla, basically, notably including bryozoans, tunicates, mollusks, sponges, cnidarians, and vertebrates, such as baleen whales and many fish, including basking sharks. So this is a property of the high density of water. It creates buoyancy for living tissues, so they're floating in the water, ready to be eaten. So all these species have in common some method of obtaining small organisms from the water, and there are many methods of doing this. For example, some kind of filtering apparatus, so feathery legs or antennae in crustaceans, baleen and cetaceans, gill rakers in sharks, tentacles and bryozoans and cnidarians to capture the food, and then some sort of adhesive, some glue, some sticky substance to retain the food particles on some part of their body prior to ingesting them. So we've got the baleen whales here, these plates of keratin that filter out krill, other small organisms in the plankton. Very efficient way of feeding high protein food items. So parallel evolution of this type of feeding in both mammals, marine mammals, and fish. So here we have whale shark with filter pads. As the water passes from its mouth into its gills, these filter pads filter out small particles, food particles in the water. Tooth morphology can be considered adapted to the type of food captured and eaten, so the sharp grasping or seizing dagger-shaped teeth of toothed whales and many carnivorous fish, such as sharks, in contrast to the crushing or chewing molars of herbivorous manatees. No sharp teeth here in the manatee. These are all basically molars. Birds do not have teeth, but have adaptations of their beak and mouth depending on the type of food they need. Puffins, for example, are seabirds that prefer small fish as food and have special adaptations in the mouth for carrying large numbers of fish back to the nest to feed young. So these include spines on the palate and the roof of the mouth, serrations on the tongue, and rosettes on the corners that allow expansion of the flesh while carrying large numbers of fish. So a puffin commonly carries 10 fish back to the nest at a time, but the record number photographed is over 60. Okay, well, that's basically our whirlwind survey of adaptations to marine conditions that are the result of natural Darwinian selection. And of course, this is just one lecture, and you can easily spend a lifetime on this subject, and people have and do. And we could do an entire course on this subject or an entire program of courses, and it would be a great program. But there are also other great subjects that we need to consider, and we'll be moving on to those next time. So we'll see you then.